There we go. I'm getting a live reading.
Yeah. Okay. So we're already like 3 minutes in, so I'll just go ahead and read the introduction. Thank you so much, everyone, for joining, and welcome to the Paper Trail. My name is Bria Macklin. I'm a postdoc at the Gladstone Institutes. The Paper Trail is an online interactive journal club that was developed by 4 former colleagues turned lifelong friends, myself, Dr. Quentin Smith, an assistant professor at UC Irvine, Michael Blatchley, a postdoctoral fellow at CU Boulder, and Franklin Hall, a graduate student at Johns Hopkins University. And so born from our love of discussing science and our yearly paper a day challenge, we came up with the idea for the Paper Trail to be a platform that allows primary authors to come and share the story behind their research. And so we really wanted the Paper Trail to stand out from other online presentations. And to do this, we came up with a set of guiding questions that prompt the author to share more than just the results from their paper. And so we're so happy that you've decided to join us for this live stream. And in true Twitch form, we ask that you use the chat feature to ask questions throughout the presentation. And so we will have moderators in the chat that will relay your messages. And Devora is really eager to answer them throughout the presentation. So please don't hold them until the end. And so if you don't see the chat feature right now, you can simply just very quickly create a Twitch login. It just takes a few seconds, and you can come back to the stream, and that will give you access to the chat feature. And so with that, I would like to introduce you to our speaker, Dr. Devora Najjar. And so Devora recently received her bachelor's in engineering and chemical engineering from the Copper Union. She recently completed her PhD. Sorry, I didn't say she recently, but she received her bachelor's from there. She recently completed her PhD in media arts and sciences at MIT, where she studied CRISPR diagnostics for resource-limited pathogen monitoring. She designed a system that she's going to tell you about today that combined a novel CRISPR assay with custom hardware to allow for biosensors that are designed for out-of-lab use. And in her free time, she enjoys teaching craft-based educational activities, cooking, reading, and ceramics. So I'm gonna let Devora and Franklin take it away.
Hi, everyone. How's it going?
Yeah, thank you, Bria, for that great introduction.
Yeah, you should be able to share your slides now.
Okay, great. So let me just choose the correct screen. Perfect. Am I sharing my slides? Can you see it?
Yeah, I see presenter view.
Oh, okay. Let's see if I can not do presenter view. And do you see the slides now?
Yep, perfect.
Great. Hi, everyone. I'm Devora Najjar. As Bria said, I just completed my PhD at MIT. I worked between the labs of Jim Collins in bioengineering and Joe Paradiso in the MIT Media Lab. And the talk that I'll give today, or the project I'll be speaking about, is a small lab on a chip project that we created that can do detection of both CRISPR or CRISPR-based detection of RNA and direct antibody detection in saliva. And first and foremost, in the spirit of the paper trail, there are obviously many people who participated in this project, and I want to make sure that I acknowledge all of them. I am going to be the representative today, but we have actually 5 co-first authors on the paper, which was something that I'm actually really proud of and really excited. I love very highly collaborative research, and I think most research is very highly collaborative, and I think to be able to acknowledge that in authorship is something that is really special. And so the co-first authors were Paul Anjali, who is a senior researcher at the Wyss Institute for biologically inspired design at Harvard. I think it's affiliated with Harvard Medical School. Sanjay is also from the Wyss, and now he's actually working at a spin-out company that works on these eRapid chips I'll speak about. Josh, who just completed his PhD, University of Bath. Elena, who is also a researcher at the Wyss. She's a postdoc in Jim Collins' lab, and myself, who was working at the Wyss while I was doing my PhD work in Jim's lab. And it's great. We work a few benches away, and so even though we were parts of different labs, the beauty of working in a place like the VEAS that has a lot of different research groups in the same location means that you can have this kind of cross collaboration on projects, which has been, I think, really fulfilling. Some of the other members on the team, Mohammed is a postdoc also at the Ingber lab. So Pawan and Sanjay are also Ingber lab members. And he is a microfluidics expert and worked with me on the microfluidic chip design that I'll speak about. Nolan was a researcher in the Ingber lab, and he now actually works with Sanjay at the spin-out company. I believe it's called Stata DX. Hani is a staff scientist at the VEAS, and he assisted with some of the pumping and the hardware that we'll be talking about within microfluidic chip. Khalid and Jonathan and she and David Walt all participated by helping give patient samples that we use for the validation of this platform. And then finally, they were the sort of mentors of the project, Joe Paradiso and Pedro and Jim Collins and Don Ingber. So a really big team across a lot of different institutions for a project that was worked on over the course of, a decent amount of time. So I'll speak a bit about this, but the project, is there a question, Franklin?
Yeah, just one quick question. So you guys had a lot of people on this team. Was this like when, in the inception of this project, everybody came together, or how did this team kind of come together to work on this?
Sure, it's a great question. I will say the two people who really spearheaded this collaboration were Elena and Pawan. I'll I guess speak about this in a bit, but there were two independent technologies that we were thinking about figuring out how to fuse together, and Pawan and Elena, Pawan on the chip side and Elena on the CRISPR side, were the ones who were working through figuring out how we can make basically put CRISPR-based nucleic acid detection on an eRapid chip. And so they were the ones who spearheaded the collaboration. I was brought in because I worked very closely with Elena and was really excited about also doing the fabrication of like an all-in-one lab on a chip-based system. And then Sanjay and Nolan work on a lot of the eRapid work in the Ingber lab. So Pawan brought them along. And Mohammed came on the team later on when we, I'll talk about it a bit later, but during the revision stage when we basically added an extra part to the paper, he came on to help. And then the patient samples, it was, I think, summer of 2020, and it was actually just quite hard to get patient samples in Massachusetts, especially in the Boston area. We're kind of fortunate to be around a lot of institutions that have a lot of hospital affiliations. But those authors were brought on basically because we had just, I think, connections in those labs, and they were able to contribute samples, specifically like blood and serum samples for the validation of the antibody stage. And then after that, we got saliva samples to validate the RNA side of the project and then revalidate the whole system altogether.
Nice. Awesome. Thank you.
Yeah. Oh, and sorry. And also Pedro, who's from the University of Bath. I believe Pawan did his postdoc with him, and that is somebody who has always been associated on the chip side of things. And so he's also just been helping out. Okay, so I guess, similar to what I was just saying, but the project actually really started in 2018. So this wasn't strictly a COVID-based project. 2018 and 2019, there were already sort of thoughts on or related to this project spearheaded by Pawan and Elena. And the goal was to try and merge two technologies. And the first technology is this project that came out of the Ingber lab, which is called the eRapid chip. And the special sauce of this chip is really just that there's a proprietary BSA glutaraldehyde coating that is drop cast onto the gold sensor surface that allows for anti-fouling. And they've tested these chips with a bunch of different additives for conductivity. So this is one of the pictures from their paper in 2019 that they had put out. And they've tested it with gold nanowires, with gold nanoparticles, with carbon nanotubes, and more recently with reduced graphene as the conductive element, so that we can use these as very, very sensitive surfaces for detection without having to worry about a lot of protein agglomeration or signal dampening due to like biofouling. And this chip is wonderful, but the primary use of this chip is really as a, or what it's typically used for is a sandwich assay. So for instance, with the project that I'll be presenting, the way that this chip works is that you drop cast and have the conductive material in that sort of gray looking piece at the bottom. And then you can just conjugate that base with an antigen of interest. And so in the case of the projects that, let's say for the COVID project, you can seed it with COVID antigens that you're interested in using for the antibodies you're trying to capture, and then you flow in your sample. The primary antibody is captured by the antigen, and then you can add things like a biotinylated antibody for detection through TMB precipitation, which causes a signal change. So you can use things like a potentiostat to get a full thermogram. Question.
Yeah, Devora, so how easy is it to attach the antigens to the surface, and does this open it up to be, you know, whatever antigens you want? Is there any limitations to the type of antigens that attach?
No, it's quite simple. I mean, they've really optimized the process to be able to do this within a day, and there is a lot of sort of drying and cleaning time, or it used to be a lot more than there is now, but you can definitely do this process in a day. And I think you can conjugate most anything you would like to in the process. A lot of it is like amine-based conjugation. And so the goal was, because we can use this for so many things, what else can we basically conjugate to the surface that would allow us to detect some other things? And can we have that be as sensitive as it can be for things that we're looking for in the sort of like biomolecule space? Can we actually also do that for nucleic acids?
Yeah, awesome. Thank you.
Sure. And so that's the sort of eRapid side of things. So a really sensitive chip that has a anti-fouling coating, quite conductive, and you can use it for lots of different things like ELISAs and assays. And we wanted to combine that with another technology called Sherlock, which came out of Feng Zheng and Jim Collins in a collaboration. And actually also at the same time, it was another version of it was also published out of the Doudna lab with basically it was funny, I think Science put them both in the same issue, and it was just like the Doudna paper, which was just for Cas12, and it talked about this specific property that Cas12 has, which is that once Cas12 and its guide find its target of interest, it will cut the target, but it also has a special secondary property, which is that once it finds its target and it cuts, it starts collaterally cleaving all of this single-stranded DNA in the vicinity. And so researchers kind of at the same time realized that this was a property of it. Cas13a was already detected, or was already used as a detection method for RNA previously. And then the Zheng lab and the Doudna lab published at the same time the Cas12's ability, and the Zheng lab also had a bunch of extra sort of like multiplexing aspects, but the actual Cas12 detection, they put them both in the same issue, which I thought was kind of cute, kind of nice. And so the goal was this very simple seeming assay for nucleic acid detection, where you can collect a sample, have a nucleic acid extraction and amplification of interest depending on the sample type that you have. You can then put in a reporter. So in the case of the sort of classic Sherlock reaction, the reporter is a quenched fluorophore that has a single-stranded DNA linker, and there it's a bunch of A's and T's. And Cas12 has been known that it's preferentially going to cleave like AT-rich sequences, so that once the Cas12 finds its target, it starts cleaving all of the quenchers off of the fluorophores, and you get this fluorescence signal that comes out of the reaction. And so the goal was, how can we combine these two systems? Is there any way for us to be able to figure out how to have nucleic acid sensing that's happening on this very sensitive electrochemical surface and have electrochemical based detection of nucleic acids? And the first projects that they were sort of testing for the system were actually Lyme disease type assays and figuring out, can we take blood samples and pull out Lyme, which it's actually quite hard because Lyme isn't really found in blood samples, but they were working on that project for a few years and figuring out what's a great application. In that process, I guess I will say, because this paper trail, there were a lot of different ways that they were trying to figure out how to do the cast-based detection. So are they going to cleave something beforehand and then flow that onto the surface? Are they going to try and cleave something on the surface of the sensor? What's the timing involved in all those processes? Like, what are the sort of pros and cons to having maybe more laboratory steps or fewer laboratory steps, longer incubation times on the sensor pad, shorter incubation times on the sensor pad? And there were a bunch of iterations of this project that they were considering. A lot of the canonical electrochemical based CRISPR papers that have been published thus far often do cleavage on the reaction surface, and it's usually like a bound like methylene blue that gets cleaved and then precipitates onto the pad. And we were hoping to try and get increased sensitivity to what's usually found in those papers, and try, especially when we're trying to go for a diagnostic level, can we get to clinical levels of sensitivity, which some of those sensors aren't able to get as sensitive as we would like. And so I'll explain more about our process when I get to the CRISPR side of things, but there was a lot of work, especially in the beginning, of trying to merge these two projects, trying to basically work out what's the best way to do the CRISPR detection, because a lot of the drop coating and casting of the actual chip, that chemistry wasn't really changing. So there was very little optimization at that point for the chip side of things. So it was really just the assay side of things that we were really trying to massage in order to make the project work. And I think a big motivation for like the pivot of this project was COVID, which it's a thing that nobody even wants to hear about anymore, but that's okay. And I mean, especially, I'm sure plenty of you on the call are involved in biology in some respects, and you know, the frustration over realizing that you have to go someplace and get your nose swabbed and wait a bunch of days just for somebody else to run a PCR reaction somewhere and tell you whether or not you have a disease that could, you know, impact you and the people around you and should be impacting your behaviors. It's really quite frustrating knowing that this, that like we have advanced so far in the laboratory, and yet that hasn't really translated in a decentralized way is like quite frustrating. Even getting things like the antigen tests that I'm showing in this image at home, that didn't even happen until December of 2020, when like the first antigen tests were approved for use. And so the timeline is kind of, you know, almost comically bad and quite disappointing. And it was something that made us, of course, think, how can we try and pivot and use these types of technologies that we've been trying to figure out a really good use for and put them together. And so the shift for this combination of the projects, instead of just saying, how can we use these chips to do nucleic acid-based detection, is really, how can we create a system that can do both nucleic acid and antibody detection at the same time? There were so many papers that came out that did CRISPR-based detection of SARS-CoV-2. There were also a lot of papers that were doing antibody-based detection, and we weren't really finding many papers that were interested in figuring out how we can do multiplexed assay testing. So on a clinical side, when you're trying to get an antibody test or a COVID test, those were two different experiences. They usually require two different sample types. Usually if you're doing antibody-based tests, they're going to draw blood. If you're doing a COVID test, you can get a nose swab, you can use your saliva, you can do a lot of things. And so we were interested, how can we figure out a way to try and get a larger sampling of things? And so that moves us into spring 2020 to about spring of 2021, which is our initial COVID experiments and optimization. And similar to what I was saying, the goal for this project was that there is a lot of information that you can get once somebody is infected with a disease, and we're usually not actually looking for all the information that can give us the full picture of the disease timeline. And so, for instance, we always are often looking for the presence of the viral RNA for an RNA virus, but that rapidly drops off after 10 days. And also we are quite interested in figuring out if somebody has developed protective antibodies, especially if you're somebody who's unsure if that what you had was COVID, if maybe you didn't get tested, or if you're somebody who's immunocompromised and is wondering whether or not you have significant levels of antibodies, or if it's been a few months since you've been infected and you're interested to know whether or not you still have protective antibodies. And so what we wanted to do for this project was figure out how can we cover this full spectrum so you get this full idea of whether or not somebody has been infected, and a more broad disease picture. And so that's a bit of what I'll talk about today. So the, I guess, sort of highlights or summaries within this work is that we developed this new CRISPR-based electrochemical assay for the chip, for the eRapid chip. We integrated it into a multiplex electrochemical chip that involved detecting for multiple antibodies of interest for SARS-CoV-2, the spike protein, the receptor binding domain, and the nucleic capsid. And we also designed a custom lab on a chip microfluidic system that would allow us to bring all these tests together.
I think one question that was asked is, what was the intended purpose after you guys started figuring out that this is going to work, that your microfluidic is coming together? Is this for a lab, or is this for like an at-home thing? Like anybody could have this or purchase this?
Yeah, that was something that we asked ourselves a lot. I think the probable intended use case for this is something that's more of like a maybe like a clinic setting. I think the system, as you can see here, it requires a bit of peristaltic pumping, and it also requires a potentiostat. And so it's one of those systems where it's like in the mid-range where it does require some equipment, but that's equipment that can be reusable assay to assay. So there's like a world in which you invest in just like the pumping and potentiostat infrastructure, and that's like put into one device. And then you have a cartridge-based system where you basically just put in different cartridges for detection of different diseases or things like that. But yeah, I think there was discussion when we were writing the paper, and I was like, we can't push this as an at-home test because that's like a bit of a stretch from what we're showing in this paper. I think that I would love for there to be a world in which this does become an at-home system, but I don't think, based on what we presented, that it's like fully ready for that yet. And there's a bit of optimization that's needed, particularly on the hardware side, because everything else is kind of fine. But I think for hardware, if we had more time, I might have like pushed to develop some more integrated systems for the hardware that would like make me feel more comfortable to claim that it can be like readily used in an at-home setting.
Right. Okay, cool.
But that's a great question. That's like definitely a big discussion for the discussion section of the paper, is like what's the sort of positioning of this. And so yeah, so similar to what I was saying for the eRapid chip, we basically worked, this mostly happened in the summer of, oh gosh, what's time anymore? Mostly the summer and fall of 2020 was a lot of the initial validation for the antibody-based testing. So this was spearheaded by the Ingber Lab, by Pawan, and Sanjay, and I think a bit also Josh. Josh also actually was doing a lot of the optimization for the CRISPR surface reactions. And they developed this wonderful multiplex chip that has VSA control, just so we can know whether or not there's any fouling issues. But we tested for the spike protein, receptor binding domain, and nucleic capsid. We chose those particular ones because nucleic capsid is like very readily present, and it's sort of one of the canonical regions within the I guess virus that you can develop antibodies against. And it develops quite a robust signal early on and can last quite long. And then for trying to see about receptor binding domain, that was interested because that's an area that's very highly mutated and mutatable within the virus. And so we were also interested in that respect, if the virus was going to mutate, whether or not you can detect for whether you have antibodies that are going to be like continue to be protective if the virus is going to mutate. And then the spike protein in general was just interesting because it can also tell you someone's vaccination status. So most people, if they have not been infected with the virus but have gotten a vaccine, let's say they should have antibodies for the spike, but they won't have antibodies for the nucleic capsid. And so there's like a lot of interesting information you can get just by having multiplex antigen detection in the first place, which was, I think, interesting, because at first you're kind of like, isn't this all telling you the same thing? But there is a lot of, I think, nuanced information you can get from antibody material that was something that was quite interesting to us. And so for these sensors, we were able to develop assays that we went through a lot of different antigens that we were testing to make sure we can have like really good antigen antibody pairs that were going to be very specific to the different antibodies of interest. And for instance, we tested IgG, IgM, IgA for all these different regions. And we found that the IgG sensors were the most sensitive and the most selective. And in fact, I would say that our test even gave us results that looked better than the ELISA results that we were getting for our samples, which was a little bit surprising, but also exciting. And I think it's tough. In general, it's like interesting to think about the, us as researchers are trying to develop newer and better tools. But you also have to benchmark, especially when you're doing patient samples, your work based off of benchmarked samples. And so it's interesting to think that ELISA, which is a system that we use to benchmark our patient samples, is something that is possibly like less sensitive and specific than something like new technology that we're trying to generate. And that's something that I think is interesting to think about, especially in the paper writing process, especially when you're getting comments from reviewers asking you to further explain your results based off of benchmarking and further like consider like why your results might have differed from benchmarked standards. And it's like quite difficult because of course you're trying to like, you have to do a lot of sanity checks, but I think it was exciting results for us and also interesting to think about when we, let's say, plotted all of our results versus the ELISA results, like looking at the two and thinking about which was, I don't know, looked like better data or was more sensitive data, and how to sort of compare those two. It was a bit interesting. But moving on, I guess. So we had sort of developed the multiplex chip that looked really great. At the same time, we were validating all of the CRISPR work for the RNA-based sensor. Yes.
Yeah, we did have a quick question on in that. How did you guys determine how many samples that you were going to run to be able to do the sanity check? Was it just availability, or was the decision-making process for that?
I think a lot of it was availability. So we were fortunate to have like over 100 serum samples that we were able to use for that. So we did about 50 to 55 for positive and negative. And I think that was a good amount for us to feel like confident. But yeah, I think especially in the early days of COVID, when let's say we were doing this work and it was summer 2020, it was not really easy to get your hands on patient samples. I think that was like huge. I mean, I think if we could have gotten more patient samples, we would have also done more testing. Like we would have run it on as many samples as we can get our hands on.
Yeah, of course. No.
Okay. And so at the same time that we were doing the antibody validation, mostly Josh was sort of spearheading the validation of the RNA system for detection. And so the goal was testing just a lot of reaction conditions. So things like cleavage time, concentration of the reporter within the CRISPR reaction to make sure we had the right amount of reporter for optimal sensitivity of the system, things like that. That was all going on. And it allowed us to develop a workflow so that when we wanted to optimize the CRISPR-based assay, we were able to have like all of the downstream work for detection worked out, which was really convenient. So that's also another positive to have a research project where you're like working on a team, because it's like everybody gets their own piece. And so, Sanjay and Pawan can be working on the antibody side, and Josh can be working on the downstream electrochemical chip optimization side for RNA, and Elena and I could be working on the CRISPR assay and the amplification and detection on our side. And everybody's sort of like working and then coming together to do the final testing of like the workflow on the chip. And so the workflow that we came up with is that we had, actually, I have to think about how to explain this in a good way because now I'm like questioning my slides. So actually, maybe I'll jump ahead and just talk a bit about the CRISPR assay, and then I'll come back to this, which is that we designed a workflow for the electrochemical CRISPR project, and it's quite similar to the original Sherlock workflow that I showed you, which is that for our side of things, after doing the blood sample and serum-based validation, we realized that blood collection was probably not like ideal overall for testing. So we pivoted to saliva samples, and then we revalidated the antibody work with patient saliva samples for our final experiments. And for the workflow for the CRISPR side of things, the goal was to collect a patient's saliva, extract the nucleic acids. In this case, it was going to be the viral RNA, and use a LAMP-based amplification protocol to amplify any of the SARS-CoV-2 present in the sample. We used a ORF1A based amplification, or like that was the region we were amplifying. And then we, instead of using a quenched fluorophore, we flowed in a biotinylated piece of single-stranded DNA. And then based on whether or not the CRISPR was present in the reaction, based on whether COVID was in the sample, if there's SARS-CoV-2 RNA present, the biotin will be cleaved. And if there was none present, the biotin would stay on the single-stranded DNA.
Yeah, one question is, were there any special considerations from a patient side of thing or technician side of thing with collecting saliva samples for your microfluidic device versus what's typically used for the PCR reaction that we, kind of use today?
What
or
do
is
you
it,
mean?
So like, sample collection right now, you kind of like spin into a conical tube and you drop it off. You just, they do their kind of work on it. Do you guys do very similar kind of processing?
Yeah.
Okay.
Yeah. I've worked with a lot of saliva at this point in my life, and it's kind of crazy. But yeah, the goal was, so I have a few lab mates who weren't working on this project, but they're actually clinicians and they're MDs. And since the early parts of the pandemic, I was sort of seeing the issues with like, especially nasopharyngeal swabs and things that require viral transport media and things like that, that there just like wasn't enough of it to go around. And so from the outset, we wanted to make sure that for our systems, we were trying to collect a patient sample that wasn't going to require any extra pieces. And so the goal for saliva was that you can, for instance, spit into a conical tube and give it to us, and then we can basically work off of that. And that was like one of the goals for the workflow.
Cool, easy workflow.
Yeah, much easier workflow, but when you're handed a tube of someone's saliva, it's like, it feels different. But it's I think I've gotten over the feeling at this point.
Was that your dream, to start your PhD and handle as much saliva as possible for 3 or 4 years?
Yeah, but I have lab mates who have done a lot of like gut microbiome stuff, and they do a lot of fecal matter samples. So I was like, you know what, in the scheme of this isn't terrible. I've seen worse. I take a peek in the freezer and I close it, and I don't want to think about it. So I think saliva is like, it's not the best and it's not the worst. And I think
lesser of two evils.
Yeah. I mean, being in biology, what a treat. What a wild ride. And so, for instance, when, let's say, SARS-CoV-2 is not detected in the system, the biotin is not cleaved off of the single-stranded DNA. So when it's flowed onto the docked PNA within the very sensitive anti-fouling pad, the single-stranded DNA will bind to the PNA. We chose PNA just because it's less charged than a DNA, and so it's just easier to grab on to the single-stranded DNA that we have. And then that biotin will then recruit a poly-HRP, and then will cause a TMB precipitation and get a signal change. Whereas if SARS-CoV-2 is detected in the system, there's no biotin present, there's no HRP binding, and there's no precipitate. And so it's actually like a signal off system, where if the SARS-CoV-2 is present, the signal is not on, and when it when it is present, it is on. When it is not present, it is not on. And so we were able to design the system and have it be very sensitive. We actually got 100% sensitivity and specificity, which was pretty crazy. We did about, I think, about 30 clinical saliva samples for this part of the project. And we actually found that this was even more sensitive than the fluorescent-based assay, slightly more sensitive. So for the fluorescent-based, we had about 2.2 copies per microliter. For this, we had 0.8 copies per microliter. And so that was something that we were very excited about, and it definitely put us in the realm of PCR level sensitivity that we were hoping for. And so that was a relief. And so at that point in the project, we were like, okay, we have these two things. They're really cool. Let's submit it to a journal. I will say, I'm not sure if I can say this publicly. I was hesitant. I was like, this doesn't feel ready to submit. These are, we did the antibody stuff, and that was based on blood samples. We did the RNA stuff, that was the saliva samples. We have a lot of workflow that goes into the saliva samples. For our initial samples, we had done spin-based purification. And so they were like, no, we got to get this out. It's great work. And I was like, okay, cool. This sounds great. I was not a co-first author at that point on the paper. I was a bit farther down, because at that point I had really only worked on the CRISPR side of things, doing the assay and optimization. So I definitely did not have a say in how or when the paper was going to be submitted. And we submitted it, and then they said, not yet. And they gave us a lot of really great feedback. And it wasn't even that it was conditionally accepted. It was simply not accepted. And we said, okay, that is totally fair. We know. And so we decided to go back into the lab. And I guess at that point, we could have decided whether we wanted to, submit somewhere else that wouldn't really care as much, or actually put the project in a packaging that made sense for making this whole system. And I was particularly excited about doing that. And so I kind of took over the project at this point for the rest of the project, and figured out how can we actually integrate these two systems into one overall system that can try and automate most of the work that goes into doing the extraction, amplification, detection on the side of the RNA, and then also allows for the antibody detection. And so that was kind of November through February, which in my mind, it felt like a very long time, because there was a lot of pressure to get this done as if it was a revision, but it feels like a very short amount of time to do a whole lot of extra work on the project. And oh wait, was there another question that I missed, Franklin?
No, you're good. I'm
Okay.
excited to go get into the
Okay, so now I'm going to talk about the microfluidic side of things, and I actually probably have to go back a bit, because the explanation that I have for the CRISPR is relevant for now. So I'll talk about this chip a bit and then go back, and then I'll come back to the side, but I don't know how to move through in a way that is going to not look bad for the screen. So I'm sorry to everybody on the Twitch. But the goal was then, okay, how can we integrate this assay? And so I worked very closely with Mohammed Yafia, who is one of the postdocs in the Ingber lab. And we spent a lot of time. This was actually also as an interesting side thing where I wouldn't talk about this usually, but for this forum, it's relevant. Collaborating with people who do not have the same expertise as you is so fun and so interesting, and it makes coming to work every day great, and you're always learning new things. But also it's funny because for me, as somebody who's, let's say, a grad student, and I'm mostly working with staff scientists or senior scientists and postdocs, I'm just oh, who am I in the scheme of things? And so I often feel as if I have to baseline defer to them. But of course, when you work in different areas and have very different skill sets, sometimes they just don't know the things that you know. And so it's important to make sure when you're collaborating, especially, to be very clear when you communicate on how and why you need certain things, especially when you're trying to spec something out. And also just don't if something feels funny or like what they say doesn't feel right, like don't assume that like they know, because they just don't have the same expertise that you do. So it's like, it's okay to, stand up for your assay. It's okay to be like, actually, this needs to be a very specific way. That's not that. And you might not know that it matters, but it really matters. And it's not like you being annoying. It's just them having a different background than you have. And like you sort of need that kind of push on all sides in order to make sure that the system is going to run properly, especially when things are as like sensitive and prone to like evaporation and contamination as things like LAMP assays. So that was another good life lesson. But we designed this very, very small chip that's a US $0.25 for scale next to the chip. And there are regions for, it's actually also, we used a high power resistor, 2 of them for heating. They're really great because they get pretty hot and they can heat very small areas, but they don't use a lot of power. And we wanted to make sure this was going to be quite a low power system. And we have reservoirs for the LAMP and for the CRISPR. We didn't publish this, but I also did experiments with this whole chip where I freeze dried and lyophilized the LAMP and the CRISPR. And we were able to run it well also with lyophilized reagents, which is great if you at some point want to turn this into a thing.
Yeah, and thinking about its current iteration, possibly future iterations for, more user-friendly applications as well. How are the temperatures kind of precisely controlled with the resistors? How challenging was that to get them to kind of maintain the proper temperatures for your biological side of things?
Oh, not bad at all. So that's also where Hanny came into the picture, was that he just did like some very minimal Arduino based programming. And so a lot of these resistors, a lot of times you're like not even going above like whatever I'm trying to think what the highest temperature we went. Maybe it was somewhere in like the 3 to 4 volts range for the specific resistors that we were using. So it's really low power. And the way that that we basically try and maintain temperature is just like PWM, like so very, very like low level programming of just basically turning these resistors on and off in order to maintain specific temperatures based on the voltage. But it just required us very early on, we just did a lot of characterization specifically within these chips where it was like Mohammed and me sort of like clipping and measuring, like filling up these little reservoirs with the specific liquid we were going to use, and just using like a tiny handheld thermistor essentially, and just trying to see based on the voltage what the actual temperature inside of the reservoir was going to be. And we use the sort of film that we use to seal these chips as well, just to try and make sure we can understand and replicate the conditions as much as possible. And so we did just a lot of like empirical testing in order to determine what the voltage would be to best maintain the temperature for each of the different reaction chambers. And then also actually while we were running a lot of the chips in the beginning iterations, it was like me standing there with like a handheld like temperature gun, just like constantly shooting at the chamber to try and make sure the temperature was being maintained and we weren't going too hot and too low. Fortunately, like there is a region of temperatures you can do with lamps. So it's like, if it's dropping a little lower and it's like 60 rather than 65, it's going to be okay. We definitely didn't want to go that much higher than, let's say, 65, so that you're not like starting to denature things. With the CRISPR, also, like if it's anywhere in the range of, like 35 to 40 is probably fine, but you also don't want to overshoot it. And I think there's a tendency to sometimes overshoot it, and then you're just like cooking it, which is really bad. But it was just a lot of like back and forth to try and make sure that we were getting the temperatures right, and very minimal, just like sort of microcontroller based programming in order to get the resistors to, let's say, do what you want them to do, which was something that I think was important to us.
I mean, briefly, do you foresee implementing any kind of thermal probes to get better feedback, kind of loop and adjustment of the temperature?
Oh, sure. Yeah. There was a separate project that I worked on before this project, which was called My Sherlock. And it was just like a very small, just RNA-based detection system. And I had a very like low level temperature sensor that was like a resistor-based temperature sensor, just to make sure that, let's say, like the incubator in that system was maintaining temperature. For this system, it's a bit harder because the area is so small that it's like really hard to figure out where you would put the temperature regulator. But I do tend to think that is probably like best case scenario to even just have like a small sort of like trip point sensor where if it's like sensing it's going over, it just shuts off. And so you can do that like with the Arduino based sensing and try and just like estimate. But yeah, I think best case scenario is like if you're building integrated hardware that can deal with this, I would love for there to be like real-time monitoring of temperature.
Okay. Well, and lastly, what considerations were made for the size of the device? How did you guys kind of come to this specific size?
Yeah, great question. Well, we had, I guess I can scroll through. We had like a lot of different iterations of the chip. It's funny because they all sort of like tend around the same system. I think for microfluidic chips, for the kind of like area in general, people are always trying to just make things as small as possible. And while I think that's great, I think also it makes things a little bit difficult. And like sometimes the smallest is not the best design. And so we did start out, if you see the top left, like the most simplistic design, or just like 2 areas for the lamp and the crisper to go to. That's sort of a hexagonal or elongated hexagonal section for where the reaction was going to take place, and then a reservoir just to put the saliva. But we basically modified it, and so the actual chip is the one that's bottom right. I asked for that square of dead space because I wanted there to be some amount of sort of isolation between the thermal heating elements and the reagents, because I didn't want them to have an issue where they're like maybe overheating just because the sample is there before it's time for them to be floated. Because I can explain more about the chip in a minute, but there's a high heat that happens with the saliva sample lysis and a proteinase K enzymatic based inactivation for the saliva. And so let's say when the saliva is going through a 95C heating, I don't want like the CRISPR to feel that because it's going to start denaturing the CRISPR enzymes. And so things like that were sort of like what dictated the design. The saliva chamber, I think I went back and forth actually a lot with Mohammed on this, because for some of the other projects I've worked on, we'll use 1 or 2 mils of saliva in order to get like a significant amount of samples such that we can detect this, the RNA in the present or RNA in the sample. This system happened to be sensitive enough that I didn't have to use that much saliva. So for the clinical samples that I used, it was about 500 microliters of saliva. And so the region was basically designed just to figure out how big does it need to be to hit 500 microliters. And I think that if we wanted to do larger samples, we basically would adjust that region, either make it a bit deeper or elongate it a bit. I think if we would elongate it more, we would probably put multiple high power resistors underneath it, just so we had enough temperature to go throughout the whole system. But that was, I think, the mentality. And then actually also the reaction chamber is this weird serpentine shape. And that was something that Mohammed and we went through a lot of iterations where the sample was just, or the reaction chamber was just flat. And it was really hard for me to detect anything from that. And I think part of it was also just like the combination of high heat and like the small region, there's like a lot of evaporation that happens. And like, I just wasn't able to hold on to like enough sample, I think, in order for it to function well. But we found with this sort of deep serpentine shape, we were able to like retain more liquid and avoid more evaporation with that sort of shape. And then also we had a lot of area for the filter membrane and a lot of area for the saliva to be running over. And so it helped both with our concentration of the RNA, and also it just helped us like keep more liquid from evaporating in the system over the course of the amplification and detection, such that we had enough material to flow over the chip.
Cool deep dive. Yeah, awesome.
Yeah, but definitely like we printed so many chips, and I'm like so grateful. Mohammed was it's kind of nuts. His wife was in Canada and he was in Boston, and she was like very heavily pregnant with twins, and he was like going back and forth between Montreal and here. And so there'd be weekends where he would print me like a ton of chips, and he'd be like, do whatever you want to do, I'm going to Montreal. and I would like work over the weekend and try and generate results. Then he would come back and I'm like, okay, this worked, this was really bad. Let's not do this again. And it was, you know, a very much similar to how a lot of collaborative projects are, like a lot of handoff. And then if he was in the lab, some days it would just be me and him sitting next to each other, like in front of like a voltage generator. And we're just like watching truly like microliters of liquid move through a system as slow as you can imagine for hours at a time, over and over again, and then just looking for the results. So it was great. But there was a lot of I mean, we could have also like designed this to death. I don't think that this microfluidic chip is designed to death. I think there's a lot more room for it to be modified, but we did probably the like just really, really rapid iteration where like even on a day-to-day basis, like we would just be changing it up. And fortunately, we have access to like great 3D printers. These were all printed on a Form 3. And also there was like sort of the limitation that Mohammed prints flat in order for him to be able to get the best amount of detail within the chip. And so one of the requirements was that the system had to be a flat chip. And that was something that I hadn't it's experienced before. And so that also, I think, limited the design in some ways where we couldn't be designing on both sides. Like for instance, for the resistive heating element, we basically designed an additional piece that like clips on. And I think in my dream world, it would just be printed to the back, but we would lose some of the surface detail because a lot of these channels are like, you know, on like the level of microns. And so we wanted to make sure that we can have super high resolution for a very small liquid flowing through, and also the detailing on these microfluidic chips. There's a lot of sort of like backstops, and like a lot of the widths of the different channels are just to make sure that we're like trying to prevent as much backflow as possible, and also having these sort of like built-in stops where if there's no pumping that's moving through, the liquid is not moving on the chip, to prevent any type of like cross-contamination, because we don't want any of these things mixing unless it's time for them to mix.
Right.
So some of the things were like built in, not going to change. So like the widths of these, actually, we did actually change the width. That's not true. A lot of the things were like not changing that much, and then some things changed a lot, which was, I think, exciting. And like, yeah, real world, like what we were dealing with was just like this every single day, which is like not, you know, it does, like, you're not going to put this in the paper. And I really wanted to, and they were like, Devora, we're not putting this in the paper.
Let
like
them know all the effort.
Yeah, it's like a lot of peristaltic pumps. It's a lot of just like, you know, microcontrollers and like chips and so many clips to keep the little, you know, things closed when they're not ready to be used. And this is just like, to me, I'm just like, this is what science looks like, and this is like what I'm used to. But yeah, I guess that's what, like, this would be good for this forum, and it wouldn't be good, let's say, for a paper forum necessarily. But just to go a bit through the workflow of the chip. So the goal is you add the saliva to the chip. So for us, in our case, we didn't heat up the saliva for the antibody detection because it's a direct assay. The antibodies actually denature in the high heat. And so, wow, the sun has set and there's no light on in my room. I'm actually just gonna go turn my light on so that I'm not just like sitting in the dark here. Okay. This might help like a tiny bit, but maybe not. And so we added the saliva to the antibody pad, and then the more saliva to the RNA detection system. And we added also proteinase K, and we incubated that at 55C for about 10-15 minutes. And then we bumped the heat up to 95C, both to inactivate the proteinase K, but also so that we were able to just hopefully inactivate whatever else there was in the saliva, be it like nucleases or inhibitors, to prevent that from impacting the downstream reaction. Because for these CRISPR-based detection assays, if you have things like nucleases present in your sample and the CRISPR hits it, those nucleases can actually by themselves activate the CRISPR system, and you don't want to have that type of false positives when you're testing these things. Never good. Oh, sorry. And so once you've done the sample preparation, we flow the saliva through the reaction chamber. Down on the reaction chamber, there's actually a PES membrane. So in previous projects, we had also tested different types of filter-based membranes for RNA concentration, and PES is just like a really great option that we had used, and it's quite well characterized in the literature. and it's hydrophilic, and it allows us to flow our material over. We use pumps that were flowing maybe at around 300 microliters per minute, something like that. So it's pretty, it's not super fast flow. And in previous experiments, we had also figured out that when you're flowing over or through these membranes, trying to keep the speed to like a mil per minute or longer. So if you try and flow faster than 1 mil per minute, you're going to be losing a bunch of the material. And so you want to try and do a slower flow over or flow through to make sure that you're actually going to concentrate the material that's necessary. And then after that, we flow the lamp mix into the reaction chamber. So the saliva is like fully pumped through. Everything is like, there's no more liquid in the chamber, but the RNA is concentrated on the PES membrane surface. We flow in the lamp mixture and have that incubate at 65 C for 30 minutes. So that's using the second resistive heater that we have. And then once that's over, we actually let it cool for like 1 to 2 minutes, just so we're not like heating up the CRISPR too hot. And then we flow the CRISPR mix into the reaction chamber. That mix actually also has a bit of a dilution in it. So LAMP is really great. It's really great at amplifying material, but it's actually so good often that if you're going to try and flow CRISPR at the concentration that we would use with other isothermal amplification methods, like something like RPA, you're going to probably also get like a false positive signal. So we actually just have like diluted CRISPR in that chamber that we flow in, just so we're actually diluting the LAMP sample so that we can have very sensitive readout. And then once that's incubated, we flow first just the saliva for the antibody test over the whole chip, and then we flow the CRISPR material over the whole chip. And then we flow in the HRP and the TMB. And then the chip is read in the potentiostat, and we get our results. And that is, I guess, these, which not very exciting to look at, but exciting when you've been like in lab for 18 hours running saliva over many, many chips, and definitely like a relief. And so our final chip was a multiplex chip that had the spike, the receptor binding domain, and the nucleic capsid, and then a PNA pad for the RNA detection. And that was all built into the chip that was going to do all the processing necessary for the saliva. And so once we got that together, I kind of like rewrote the whole paper, which is fine. I think that happens. And then we submitted it and just got responses, I guess, here. In April, we just got responses from the reviewers, but it didn't require any extra experiments, which is also like always a relief. We resubmitted, and then the paper came out, and that was really exciting. And I think if I think about next steps for this work, I think it would be great. These systems are not very quantitative, they're mostly qualitative. A lot of detection methods that use amplification as a step prior to detection are rarely going to be quantitative. So it'd be great to have systems that can build in more semi-quantitation or quantitation. These electrochemical sensors have been reused previously for the antibody-based systems, but we haven't tested out reusability for the RNA and the PNA-based sensing. So it would be great to test that out and see whether or not these things can be recharged and reused. And then also just developing more microfluidic infrastructure and automation for latent monitoring, whether it be like in somebody's home or even for, I'm really interested in using these things for environmental monitoring. So figuring out like, can you set up a system that you can put out in the field that can be doing latent monitoring? And that's really it. I guess we kind of kept to time, which is good.
Perfect. Yeah, right on time. Excellent. Yeah, at any point right now, there are any last minute questions from the chat, let us know. And we really appreciate you coming on. It's a really interesting paper. Interesting how you guys, had two separate things, how to put them together as a microfluidic device in a really quick turnaround. I mean, that's fast. That's rapid.
Yeah, sorry, I was here just listening, of course. But yeah, I mean, I think that was the most amazing thing, is that you said at the beginning you weren't leading this, and then, you guys kind of submitted, got that rejection, and then it became, okay, how can we revamp and combine these things? And then you took over from there, and in such a short amount of time. I think that's really, really impressive and very exciting, the work that you guys were able to do. And yeah, like you said, just having access to that 3D printer where you can just rapidly print off new designs and test them right there is key to the success of this. So that's really amazing.
Yeah, I will say that the VS has a lot of fabrication facilities, and I think if we didn't have access to 3D printers and things like laser cutters for the PS filters that we can use kind of like every day if needed, it would be so much harder to be able to work really quickly. And so there is so much just on like the materials and facility sides of things of like, do you even have what you need? Like I had a lot of things around already. So doing things like rapid iteration just became so much easier. And also I think every CRISPR project, or sorry, not CRISPR, every COVID project, it just felt like, you had to get out as fast as possible. And so there's just like a speed that we, worked out for this project that I think like wouldn't have existed for any other type of thing. And I think definitely it was like a huge motivator to try and just like throughout the entire pandemic, just like work as fast as possible. And I think that time frame is kind of nuts, but also I don't know, sometimes you just do it and you're like,
Yeah.
this is crazy, but I'm just gonna do it. And then but that's also like research is like intense periods of like huge amounts of lab time when you're like, because like once you're getting the good data, you're like, we got to keep going. Like I have to just get all of it as fast as possible and hope that like nothing changes so that like it can all look good and consistent. And then you go into more of like a latent period of like you're either writing or you're doing figure designs. I did all the figures for the paper and that like took a bunch of time also. And yeah, like it's definitely like it's research in spurts, and I got lucky that I was able to have that like spurt of time and like also the involvement of other people so that like it could happen. But it was definitely it was able to go so fast because it was built on so much else, which I think often doesn't get acknowledged.
Yeah, no, like you said, it's great to acknowledge like that there was a platform there before and you could take
Yeah.
off from there.
Yeah.
I want to thank everybody for joining this stream. I'm gonna end the stream now. I forgot how to do that. Hold on. Here we go.